Hi, I'm Shantanu, a senior policy researcher at QTCI. I am joined by two fantastic speakers today, Deepak and Kiran, working at the intersection of quantum and biosciences. Uh, as a start, I would like each of the speakers to introduce themselves, the problem they're working on, and how they got started at the intersection of quantum and biosciences. Deepak, do you want to start?
Oh yeah, wonderful. Thanks a lot for this invitation, and it's wonderful to be here and share what we are doing, as well as try to see if we can motivate more in terms of community building activity around quantum sensing and life sciences. So just my background, myself, I'm a mechanical engineer, and then I did my PhD in biomedical sciences for 15 years. I'm in the business of high tech and deep tech equipments. Precision measurement devices here. So in the past 2 years, I think it's almost 24 months now that we co-founded QD Sense, largely with the mindset to translate whatever was in the physics lab into a product that life science community can consume and create more value in that area. Yeah. I'm very excited about it, and some of things that we are really proud of is within 24 months we raised close to 12 million in dilutive and non-dilutive funds, and we have around 5 equipments deployed in the market, and there's a lot of learning in terms of user experience and also fidelity of the device. And just recently, we were able to crack a problem, muscular dystrophy. And that's something we're very proud of. And maybe I'll share more about how quantum sensing can help in neurodegenerative disease and the value that truly society can capture with that. Yeah, thanks.
Yeah, thank you, Deepak, for that introduction. And congratulations on cracking that problem. I think we'll talk about it more during this session. Kiran, over to you.
Well, hi everyone. My name is Kiran. I'm a chemist by training. Did my PhD in organic chemistry, then dabbled around in biology for a while, then worked in big pharma, and recently joined Quantum Biosciences as one of their scientists leading R&D for them. At Quantum Biosciences, we're primarily interested in predicting organ injury before it actually happens. And since this is something that needs to be done at a very early stage, you need to be able to pick up markers that are very early stage. And that's how we intersect with quantum sensing. We use nanodiamonds, we use NV centers for picking up those earliest signals of stress in cells. And we use that data to ultimately predict whether an organ is going to have failure or not, right? Happy to talk more about what we do, how we do as we go along in this panel. We are currently incubated at C-Camp in Bangalore. We have just raised our first pre-seed from the National Quantum Mission. We're building the team together. We're putting some experiments together. So we're very, very excited. Thank you for having us here.
Yeah, thank you, Kiran, for that introduction. Do you want to continue and walk us through about how and why quantum sensing is being utilized, and what's the difference between classical sensing and what are the limitations that brought about you looking into quantum sensing in the first place?
Yeah. So for the particular problem that we are addressing, we are looking at a very fleeting chemical molecule that forms within cells, generally around the mitochondrial area. And these sort of molecules are what are generally umbrella termed as reactive oxygen species. For a chemist, these are typically what are called free radicals. They are molecules that have an unpaired electron in them. And by virtue of having an unpaired electron, these molecules are inherently unstable, right? So if you use classical methods, there is no good way to detect these species. So you would have to have a reaction that is set up where you consume this species, and that's the classical way of determining how much of these raw species you have. It does not give you a way to do real-time sensing, does not do a good way of giving you tiny fleeting amounts of these raw molecules being formed, right? And that's why you need alternative techniques. You need newer techniques that do not consume ROS, technique where you're just hanging around by the side of ROS while ROS is doing its job, and you're still able to detect ROS moving around you. That's the sort of technique that we are looking at. And that's something that we are able to do with quantum sensing. In this particular case, we use NV centers, and then these NV centers are sensitive to any sort of change in magnetic field around them. And I told you free radicals have an unpaired electron, which means they behave like tiny magnets. As they keep moving around this NV center, they create noises, and these noises we are able to pick up, right? And that's the technique that we have. Any classical technique would essentially require huge amounts of sample to quantify this sort of change, or it would consume the ROS, or it would change the way the cell functions. Whereas with this sort of a technique, it allows you to just sit there without interfering. I think that's the beauty of the technique.
Yeah, yeah. Exactly. In that line of thought, the whole idea about QT-Sense, how we came up is to basically target this most complex short-lived species, and not only reactive oxygen species, because quantum sensing, if you look at the NV center, NV sensing, NV means nitrogen vacancy sensing method, especially NV negative. And if you see that it can pick up any magnetic noise around it, and reactive oxygen species has as an unpaired electron, but there are many paramagnetic species inside the cell. It could be metal ions, which has more unpaired electrons, and they have maybe a better magnetic noise that our senses could be more sensitive to it. So, for example, you talk about Fe2 plus, which plays a very important role in Fentine reaction and leading to ferroptosis. So are you able to map that particular magnetic ion's behavior in a living cell? So I'm just adding on top of what Kiran just mentioned. So overall, one should look at quantum sensing, the way the philosophy of QT-Sense, how we see this is in this way. There are many techniques that has revolutionized biology as such. So, for example, if you look at genomics, it basically tells you what could happen, and there you look at mapping out. We have the human atlas being built around the very much in terms of DNA mapping there. And then you also have proteomics. Proteomics is designed around, what is kind of, is happening. But none of these two building blocks of biology, genomics and proteomics, are able to provide the functional aspects inside a cell, and this is exactly where, we call it a radical omics. So basically what it means is that we can map free radicals inside a living cell or a tissue or an organelle or a fluid, and by doing so we're going to give functional information to them. It's not a static snapshot, that genomics and proteomics, but we're going to give functional information. And using our Quantum Nova platform, one can extract this, and what it can in terms of insights, what it gives is it tells you why the cell is phasing out, and at what time point it phases out, how much of stress it has in its real conditions, and then what happens to the stress state when you trigger them. So all of this information is very important at functional level. The dynamic information has its own beauty of solving problems compared to static snapshot. So I think quantum sensing, Elda, now come to the technology. Without quantum sensing, I wouldn't believe there would be the third omics coming in like this, free radical omics. This is largely because the beauty of seeing invisible, something that is so invisible, so difficult in terms of, here we're looking at ultra low magnetic noise. Basically, at the end of the day, it's sensing ultra low magnetic noise. It could come from single unpaired electron, or it could have it more than one unpaired electrons in a species. There's something that one should understand, is that within free radicals we have different categories. You have superoxides, you have hydroxyls, and then you have nitriles. So the most important part is that they all have such short lives. For example, hydroxyls have a nanosecond short life cycle. not life cycle, the shelf life. And then every cell has something around, they go through 10 power 11 times free radical cycle per day. A cell can go through 10 power 11 times free radical cycle per day, but there was no method directly to monitor it. Indirectly you can have label method, you can react to form peroxides and see the fluorescence and stuff, but direct representation of free radicals was not possible without quantum sensing. So seeing something invisible is what quantum sensing enables, and I think that's going to drive a whole different discovery within biology.
Yeah, what I'm hearing is it also allows us to look at the samples dynamically and also provide a greater resolution. Is that a great representation of what's happening within the quantum sensing space, or do you see there like some trade-offs happening regarding the sensitivity that we're trying to achieve?
I think there are two different things in my opinion. One, what you achieve is a spatial and temporal resolution, which is really far better. When I say spatial resolution, we're talking about subcellular resolution inside a cell, and targeting different organelles, like mitochondria and the endoplasmic reticulum. You have nuclei. For example, one could understand between endoplasmic reticulum, mitochondria, how they communicate, how the calcium influx happens. That's a big hit in terms of mapping physiology at subcellular level. That's something that's been provided by quantum sensing, because these NV-centered fluorescence nanodiamonds are around 70 nanometers in size, so it's beautiful that we could have uptake and then target them to any organelle. And we recently also demonstrated that we can target not just membrane-based organelles, we can also target membrane-less organelles, and this is where the new areas is going to be built in another way, which was not possible, I think, prior to quantum sensing. So I think spatial and temporal resolutions are very important that quantum sensing enables.
And Kiran, in your work, how do you look at the bigger picture of like isolation of so many magnetic noises, and when you're going to the diagnostic part of it, how do you target a particular free radical group, as you said?
Yeah, no, it goes back to what Deepak was saying about having spatial and temporal resolution, right? So it comes down to that. Think of it like you're trying to measure pollution within a city. Now a city is just a massive space, right? And your representative sample for measuring, let's say, pollution, either AQI or maybe you're measuring sound level, you now have to describe where you're going to measure that. Are you measuring it right next to a freeway? Are you measuring it in a residential area? Are you measuring it in an industrial area? It works out because the city is diverse in its shape. It's massive in its shape. Now with quantum sensing, you can have the same dynamics of the city to start actually playing within a cell, right? Deepak told you that these sensors are about 70 nanometers, and you take a typical cell which is about thousands of times bigger than that 70 nanometer size, which means where you are actually getting this data can now give you localized information, and you're not getting gross cellular level information, right? So if you use the classical methods, you would get gross cellular level information. You get fluorescence across the cell, you would get fluorescence outside the cell. Those are the sort of data that you would get. But now with these really tiny sensors, you can say, hey, I'm getting this data around mitochondria, I'm getting data around the nuclei, I'm getting data just outside the cell membrane. And the dynamic range that you're talking about around your sensor is maybe about 10 to 20 nanometers around it. So any noise or any paramagnetic noise that's around this 10 to 20 nanometers around the diamond is what you're actually picking up, right? So you get this hyper local data that you have, and so from it itself you get some sort of resolution. So now you know that around the mitochondria is where all your superoxides are going to come out, and then you wait a little longer, go a little further into the cytoplasm, these superoxides are going to get converted to peroxides, and then further downstream into something else. So The closer you are to a mitochondria, you're likely to pick up superoxides, right? Maybe the closer you are to an endosome, you're probably going to pick up a different type of radical species. You're closer to an endoplasmic reticulum, you're going to pick up noise from different species. So one of the things that we're trying to do at Quantum Biosciences is, can I pick up signatures for different types of radicals that we are seeing within the cell? So can I find specific type of relaxation curves that will correlate with superoxide species? And is that relaxation curve looking different from the relaxation curve of a hydroxyl species? Is that looking different from a peroxynitrite species? So there are about 7 to 8 different sort of these species. One of the questions that we are asking is, can we differentiate from the relaxation curves? And how is that going to make a difference is ultimately when we tag it to a specific disease system that we are looking at, we should be able to tell, oh, this sort of an injury or this sort of a stress gave rise to, let's say, a bump in superoxide signature, or some other type of stress in the cell gave rise to a peroxynitrite bump, right? So can we tell that from these quantum sensors is something that we're actively doing at Quantum Biosciences. We're hopeful at some point that we will reach a sensitive enough way to detect them in good confidence. But right now, it's too early to say if we will be able to do it.
We already have some of the questions coming in regarding what kind of filtering techniques are being used and how are these sensors manufactured. Do any of the speakers want to go in a bit more detail regarding?
What's the question?
Go ahead, Deepak.
No, sorry, Karan, go ahead. I was just looking which question is that then. Shantanu, what's the question? Is like exactly, is there a question that pops up here that I can say?
Yeah, it's regarding what kind of filtering techniques are used to for magnetic noise that is produced in the body, and how do you manufacture and acquire these kind of sensors?
Right. No, yeah. So what do you filter it out? I don't know what filtering out means exactly, but the NV negatives are basically, they convert magnetic noise into optical signal, basically, right? So that's the quantumness in the sensor. You basically NV negatives are they have 3 spin states on their valence electron, and then you can polarize them, and then using any of the technique in our platform that we sell, we basically polarize them using laser pulsing technique. One could also use microwaves for that. And when you polarize them, and when they tend to fall back to its equilibrium state, then you basically it basically senses the magnetic noise, and the rate of the decay would change when it tends to fall. And that rate of decay gives rise to red fluorescence, and this is what we basically do at single photon detection level, and then we map that relaxation curve here. So filters and stuff, we have, this is 532 nanometer laser excitation, so we have 650 nanometer filters, but you could use different filter sets. That's not that important depending on what fluorescence you're looking at. For us, we do 630, 650 nanometers filter set at the detection level. In terms of the sensor itself, there are many suppliers of the sensors. We don't produce the sensing, the NV sensors ourselves. We qualify them. We qualify them with respect to photon count. We qualify them with respect to distribution. We qualify them with respect to ability to functionalize it. They, for example, typical NV sensors should have 5 ppms of nitrogen, let's say, or 10 ppm of nitrogen. So we have some criteria as how we select those sensors, and then we offer it to our users as a consumable, basically here. Yeah. So, but if you look at the classical method, it's a high temperature, high pressure method. You do, create those in nanodiamonds, and then you use ball milling to crush them into small pieces. There is still standardization being put into the production process. So Yeah, but we basically qualify them, what we get.
Yeah,
Um,
I can just elaborate a little bit. I mean, coming to the fact that there is so much magnetic noise within a cell, um, like the typical way to do everything in biology is to have the right set of controls, right? The assumption is that over the two samples, if you have the right sort of control, the noise that's in the background without that change is essentially the same, right? So by having the right sort of controls, you automatically set up a way to differentiate between magnetic noise post stress and pre stress, and that in itself should be good enough for you to pinpoint and say, okay, this the change that I'm seeing is attributable to this, right? There is no other way to filter out the noise, because you can imagine every molecule that's around this NV center, that's just by virtue of its movement, it's tumbling, it's rotating, it's vibrating. All these sort of movements exist around the NV diamond, and you cannot, in principle, you cannot really tease out every single of that movement and then start filtering it out. So it's not going to be a trivial task to filter out. But what you can always do with biology is you can have a pre and a post, and ask the question: is the change that I'm seeing, is that correlatable with the stress that I put in? And if that's the case, that's how you would filter out.
and as these technologies are moving from lab to commercialization, as you have been going, um, how do you see these techniques fitting into the pipeline? Uh, is it required to fit in the existing either diagnostic or research pipeline? Are there existing tools that you're building on top of, or you're creating a separate hardware that would need its own pipeline because you're providing more information, more data, which needs to be processed earlier?
Yeah, I think it'll take some time for us to reach a sort of scenario where we think of these techniques being translated to the clinic, like how you have blood tests today, right?
Mm.
It'll take some time for it to reach that same sort of maturity that we usually think of. Okay, there's a take someone's sample, take that blood sample, put it in a machine. The machine does everything. We are still a little away from that. What we are doing at Quantum Biosciences is we are setting ourselves up in a path that leads to that process. So you essentially today have an instrument that is primarily used in a super specific R&D setup, trying to convert that instrument from just having this R&D scale throughput into something that ultimately needs clinical scale throughput, right? That requires a lot of engineering effort, and that's the path that we are on. Obviously, for all that to happen, a lot of science has to work out. There has to be the right sort of diamonds that are being made. The diamonds have to be made in very specific manners. That technique is something that's still evolving over time. If you take the diamonds that are currently available, they are not yet ready to go into clinic. They have a lot of variability in them. It'll take some more years until those techniques get fixed, right? So that's in the very core of the sensor itself, is the ability to have diamonds that consistently perform in the same way. They're consistently sized, they're consistently oriented, and all that. Apart from that core quantum sensing unit, the rest of the stuff falls into the regular pathway of a medical diagnostic device, right? So you need to go through regulation, you need to go through fidelity, you need to go through robustness, you need to go through ruggedization. All that process still has to happen. Deepak could also tell you about some of their efforts in trying to make this instrument a lot higher throughput. So yeah.
Yeah, no, indeed. What Kiran mentioned, that there are certain challenges, and just to give a few examples of it, the systems that we put in the market, they're largely, all of them are for research use only. They're not for diagnostic purpose. And within the scope of research use only, we picked up a few clinical studies. One of them is sepsis, for example. There's an ongoing clinical trial at the moment in the University Medical Center nearby our facility here. So there, what we are doing is quite interesting. For those who don't know sepsis is basically kind of inflammation that leads to organ dysfunction. It's not just the bacterial infection in the blood. It has more than that, per definition. What we did was, at least the standardized protocol is, we take the blood samples of those getting into admitted to ICU, and then we extract the neutrophil from the blood, and then there are many types of neutrophils, but you need to be very careful with the protocol that these neutrophils don't get activated when you extract them, and then study its free radical load, the behavior or the concentration of free radical in these neutrophils. So you take a blood sample, you extract the neutrophil. In that soup, we are able to map out the free radical concentration. And out of 30, and I think now it's around 60 plus data sets we have on this topic, we are able to detect sepsis earlier than the classical SOFA score. It's kind of organ failure analysis that a clinician would do. As soon as you walk in, you have sepsis or not, the first thing he looks at your blood pressure. There are many other methods that are objective, and they give a score called SOFA score, and if the score is high, then they admit you as a sepsis patient. Sepsis is very complex and the most rapid towards mortality, because every hour you delay in sepsis diagnostics increases the mortality rate by a certain percentage. So that's one of the area where we are exploring there, as Kiran mentioned as well. How do we get that data reproducibility and repeatability by managing those biological variations? So there is a lot of work to be done in that space there. But in terms of clinical work that's going on, there's also other project that we are working on in terms of clinical trials, especially in the area of tumors. So you have when you do biopsies, you get some idea about the perception of the tumor. Especially, let's take an example. If you do a mammogram, you get the bright spots, and you don't know if the bright spots means more fat or is it tumor. And so there is a way that we were asked to look into that from quantum sensing, and we have made significant progress in that area to help some of those people who use the kind of conventional microscopy or any other technique to identify tumors. So all in all, we are touching different fields. A lot of elements within the instrumentation has to be standardized, sensor has to be standardized, protocol has to be standardized. I think, in my opinion, within 2 to 3 years, there'll be somebody in the market who will come out with a diagnostic tool. And I wouldn't say it's us. We see our trajectories is a little bit different than diagnostics. We cater to the needs of drug discovery. All the work we put in is how can we improve the fidelity of the quantum sensing microscope that we are designing and deploying, and we are looking at how can we use this to build multiple in vitro disease models that that the drug discovery team could use. And also, we have recently seeing a bit of traction in the AI field, because a lot of the data that is being fed to AI is largely static snapshots. And if you try to provide an in vitro disease model that is highly functional and dynamic in nature, they start to see a different, outcomes in some cases, especially in rare diseases. There is some things happening there. So not diagnostics, but largely drug discovery and academic research as well, and build more in vitro disease models that the market can consume.
So the challenges and bottlenecks you face for each of, I'm guessing for drug discovery versus diagnostics would be different. So the bottlenecks, are they more hardware oriented right now, signal processing, or the software implementation itself? What kind of challenges are you facing, or are they equally important? Or is it more of there's lack of data and tests being run that you're like slowly building towards? on where the applications are.
No,
You
from
want
the
me to take?
instrumentation. Go
Go
ahead,
ahead.
Kiran. From the
Yeah, so-
instrumentation side, on the system architecture itself, um, I don't know. I think it's going to get standardized at a point, and we see that already happening in our own ecosystem. What we do is we have a confocal microscopy on top of confocal microscopy that we have designed. We are adding this quantum sensing module into it. And confocal microscopy is not new. It's there for decades. So we are just adding on top of it the quantum sensing module to it. What would happen, I think, is multiplexing will emerge in that area very soon. We already start to see that multiplexing opportunities. Also, make the signal when you talk about challenges and system architectures, making it high throughput screening. So at the moment, most of the measurements are single cell, because then this is a single cell measure technique what we have, and there we are using a petri dish, and then and if you look at drug discovery or any of them who are at the preclinical stage, they always want to screen more compounds. So high throughput screening, high content screening is going to dominate quite a bit there. And there things will get more complicated than what I defined earlier. It's just a confocal microscope, add quantum sensing model on top of it. Okay, that's fine. It serves the basic requirements. But when you want to make it faster, those measurements, then the whole data acquisition system, the time to insight pathways, they are going to get complex in that area. And we see it already in our ecosystem, in our company, when we are already trying to achieve that. Yeah. It's important to look into that. Yeah.
Kiran, you want to add something?
Yeah, I think Deepak did touch about it. Currently, the technique that we have, where we're using quantum sensing uh, to detect radicals, right? You're essentially detecting a chemical species within a biological system with a very sophisticated physics instrument, and you're doing that at a single cell level. And if you're going to do at a single cell level, you're not going to get a good representation of your entire sample. You have thousands and thousands of cells in your sample. And if you're doing detection of one diamond within one cell at a time, you're spending X number of minutes getting that data out. Then the question is, you can't spend times 1,000 of that for your single sample, and then multiply that by the number of samples. So you can imagine how slow the process can actually become. So at some point, there will be a trade off between how sensitive do we want our measurement to be versus how quick we want it to be, right? We are yet at a stage where we are still worried about, okay, I want the most sensitive measurement right now. I'm not really worried about being high throughput at this time. I just want to make sure that the science works. I want to make sure that I'm able to detect these absolutely hard to detect chemical species out here, which there's no other way to detect it. I'm at least able to detect it, right? Once the community feels like, okay, now we have reached a collective agreement that yes, we are able to do this in a reliable manner. This works. There is a disease correlation here. Now we have to do this faster. That's when again there will be a little bit of a revolution that would be required in terms of instrumentation, in terms of the physics of the whole thing, of how do you detect across multiple cells, multiple populations of cells, multiple diamonds, and so on. So today I would think the bottleneck is basically that, do we know that this can become high throughput is the entire question. Okay.
So, for as for the timeline, when do you reckon there would be some commercial applications of this already? I think Deepak mentioned like 2 years down the line, we start seeing some of the applications actually
Yeah.
being commercialized. Is there a similar timeline that you put it in?
Yeah, I mean that's the hope that we are working on. That 2-3 years down the line, we should have a commercial version that works out in the clinic, right? I'll just dive a little deeper into what exactly we plan to do. So, like I told you earlier, that we want to be the company that basically predicts organ injury before it happens, right? And the way we want to look at it is before a cell or before an organ ultimately has damage, it has to shout out saying that it is under stress, right? And the way that it shows up stress is essentially in these reactive oxygen species. Comes down to the same free radicals that I spoke about, which are present in fleeting amounts. They come in and they go off. And until now, we didn't have a technique. So we have a technique that we can detect these. That means we are essentially now able to interrogate whether a cell is stressed. If a cell is stressed, can I correlate that stress into ultimate damage? Right. So that's the pathway that we are on. And you need a starting point for this whole thing. And for us, the starting point is basically acute kidney injury. So acute kidney injury occurs usually in major surgeries. So on an average, you have about 300 million surgeries, major surgeries that happen in the world every year, right? Which is, if you think about it, it's a ridiculous number of major surgeries that happen. And approximately 20% of them usually end up with acute kidney injury, right? So you're talking about 60 million-ish people who will have acute kidney injury. And then imagine these 300 million people go into surgery and 60 million of them are likely to have AKI, but there's no way today to tell you that you're going to have AKI, right? And the only way to know that you have AKI is to look for a marker called creatinine. Anybody who has got blood tests done, creatinine is something that you always look for in your either serum level or you look for it in your urine, but it usually shows up 2 to 3 days after your kidney has got damaged, right? So the question that we are asking is, instead of waiting for 2 to 3 days and getting to know, okay, creatinine is increased now, that means my kidney is already damaged. Can I tell you 2 to 3 days before the kidney even gets damaged? Can I tell you that this kidney is going to have AKI, right? And how can I do that? By looking at stress markers. And I'm able to look at those stress markers with quantum sensing, right? So that's the whole paradigm that we are working on. We work with the instrument that Deepak's team has designed. That's our baseline instrument. We hope that we will be able to generate clinical trial data that will support the whole argument that we can look for ROS data very early on at very early time points and able to predict AKI, right? So that's the pathway. And if that pathway works out, ultimately it will come up with a clinical diagnostic equipment where you could have a microfluidic chip sample. You take a blood sample, put in a microfluidic chip, and then push this cartridge into a machine, run a quantum sensing experiment, tell me what's happening to ROS signature. So that's the hope. And we hope that we should be able to see some sort of early signs of commercialization in the next 2 to 3 years for that.
That's fantastic. When I think the product moves to a clinical setting, there's also like size, weight, power, and cost requirement that comes in. And from what I'm hearing, 2 years down the line, a lot of these challenges would be resolved. Is that a correct situation assessment answer?
I think if you look at the baseline instrument that we have, which is QT Sense design, you will see that in terms of these, it's not really very far off from instruments that are out there already, right? It does occupy a little bit of real estate space, but really apart from that, I don't think it's not consuming amount of power that is ridiculous. It's perfectly handleable in terms of the way it is, but I guess some sort of miniaturization and ruggedization is going to happen. That's inevitable. Yeah, I mean, I let Deepak talk a little more about how the instrumentation takes shape.
Exactly. I mean, the one of the if you go to diagnostics, there's more about there's a lot about at least in Europe we have the MDR directives and so on and so forth. There's a lot of regulatory things you need to take into account. For example, you can't use the conventional USB cables in your device. If you want to have an MDR CE certification and so on and so forth. So there are certain regulatory aspects we need to take into account. But there will be a lot of improvements if you want to go to diagnostics area. One has to do for just a simple, no-brainer example would be that if I draw the blood, in a tube, I don't want to open that tube and then deploy it in a petri dish. I want to be able to measure it in that tube itself, the free radical concentration. So how can we make that seamless transition into a device, that at the moment consumes a petri dish? So there are ways that one should be able to map out the reactive oxygen species or the stress level in a blood sample without with still keeping it sterile. So we call it as on the bedside devices, so that it becomes a companion diagnostic. So there are quite a bit of challenges there, I think, but this is something that experts like QBS, they can prepare those unmet needs, and we do the instrumentation if necessary, or they can add on the topic, which as Kiran explained about microfluidics and so on and so forth. But there is also, I would like to mention that beyond clinical, in terms of commercial aspects, I think of all the quantum, if you talk about quantum computing, quantum communications, quantum sensing is a low-hanging fruit in terms of impact in society. This is always my take, and this is what we are being we demonstrate this by example. Currently we have bookings of around 11 devices in a matter of 24 months. So we have revenues coming in within the quantum world. That's quite strange. Usually you have been backed by heavy investors, or you have basically launch customers who are largely in defense, who are throwing in money. So here we are seeing that the market is asking for a device like this that they want to use. For example, I still want to focus that in drug discovery, there's a lot happening where we can enable that simple. We just recently came across a case where they wanted to explain how a drug that they developed could target this signal pathway, the stress pathway in a cell. Either they want to activate that pathway or they want to inhibit that pathway. So in your cell, just a little bit of biology is that these free radicals are there all the time. It's just that when it goes out of balance, you get the stress. And free radicals are good in your body because they do signaling. They tell you, hey, you will have this inflammation, so that it activates certain antioxidant genes. So they're good to have, but if they go out of balance, then it becomes risk to a disease, basically, right? So there are certain compounds that is being developed at the moment that they want to target this pathway. Now, if they want to target this pathway, they need to have a reliable measurement technique to target that pathway, because the conventional targets are always based on, okay, we measure the reaction of the free radicals. They have very low photostability. There is a lot of infidelity in the existing methods. Now with quantum sensing, when you can directly measure these free radicals, then the entire farm, at least most of the drug discovery teams that we are engaging, they get excited and they say, hey, you know what, why don't I target this pathway? Or I have targeted this pathway and I have failed. With your technique, maybe I can get more insight to my company. One should realize that the whole drug discovery and development is $1 billion, almost a decade-long business, over here. And what we are looking at in terms of commercialization, where we have been partly successful in a short period of time, there is we are able to remove the weakest compound from the preclinical research entering the phase trials. So that's fail fast, fail cheap is where we are positioning quantum sensing in drug discovery, and we have built quite sufficient data in oncology and neurodegenerative diseases to help these pharma companies. And we also realize that it's not just drug discovery in the pharma. The same disease models and targets that we are, and controls, and so on and so forth, the whole protocol that we have been able to demonstrate on our device is also being consumed in spatial biology within academic groups as well. some of the top-notch spatial biology groups, some of them in redox biology, they are now able to use our platforms to build more discovery and maybe high impact publications there. So we are seeing drug discovery and some of these top-notch universities, using our platforms to create value there. Yeah. Apart from diagnostics topic, yeah, and clinical topic. Yeah.
Yeah, these are like definitely very exciting times. Going into some non-technical areas, I would like to ask two questions. One is the processes that you described, are these applicable only to human cells, or is research applicable directly to plant-based cells as well, where they can be utilized for uh, figuring out agriculture diseases? And second is, what are some less obvious use cases and alternative use cases that you've like come across research, either like combination of technologies or something that needs to be that has received less attention, but you think deserves more? Kiran, do you want to take that question first?
Yeah, I mean, it's. I think the amount of work that's done in humans, or rather human cells, with quantum sensing is still in its early stages, right? So whatever data that we have primarily seen, they're all mostly from animal models that we have seen. I don't particularly recollect if anybody has done this on plant cells. I'm just not in touch with plant biology. Maybe Deepak knows if there are some applications in plant biology, but I would uh, I would think there are some challenges. Ultimately the NV sensor or the quantum sensor that we are talking about needs to be picked up by the cell, right? If you need an intracellular measurement, you need it to be picked up within the cell. Uh, Plant cells, by virtue, usually do not pick up stuff, right? Because they usually will have a cell wall around them. But there could be definitely some subcategory of cells which do pick these up. There are ways in which you can bypass this, like you have these little fungi called yeast, and you can't push diamond sensors into them, but you can measure what's happening within the cell by having the sensors outside the cell, right? So they're sensitive enough to pick up what's happening inside by still staying outside. I don't know if people have done that with plant cells, but most of the data that exists, most of what we know today exists primarily from mammalian cell lines. And as Deepak mentioned earlier, there's also human blood data that we are seeing. There are now some amount of organ level data that we are seeing. We are seeing organ slices into which diamonds have been perfused in. They've been taken up by these cells, and you started getting some data for early organ level data. But then there is an inevitable limitation to the technique. It's an optical technique at the end of the day, right? So you have to shine light and you have to pick up light, which means your medium has to be transparent at the end of the day. You can't have these sensors deep inside your liver, right? And you can't have a scanner that's outside that's picking that data up, right? So the technique today is not amenable for that. It's amenable primarily for ex vivo work, which means take something out of the body, do an experiment, and then study it. That's what it's most amenable for right now. So that's the answer to the first part of your question. I forget what your second part of the question was.
Some less obvious use cases that you're
Oh,
excited
yes.
about.
Yeah, I think Deepak touched a little bit on the fact that people are a lot more excited about drug discovery at this point, right? You're again there. I think the limitation is just about being high throughput at this point. Once that problem is fixed, I think drug discovery will definitely lap it up because it gives you very instantaneous readings to what's happening every time a molecule interacts with a protein within the cell. Is there a localized change? Is there a temporally resolvable change? And if you're able to pick that up with this quantum sensing, definitely there is a lot that can be done in drug discovery space. I think there can also be things that we don't yet understand, right? Like, let's say somebody travels deep underwater, and you want to come back up and you want to see what's the sort of stress level in their cells. Or somebody is thinking of space travel, for example, and you want to think of, okay, what's happening to cellular stress if you're doing space travel. So these are a whole lot of things that kind of open up, because we're able to get this sort of fine-grained data that you were never been able to get until now. So a lot of these applications we don't really know, they may just come up as technology gets better and better. Yeah.
One of the things to add is that with the two things. One, the less obvious. I think I'll circle back to another thing. So instead of plant cell, I haven't come across an application there, but there are cells, there are cell lines, there are mammalian cells, primary cells, whatever. There is also tissues. As Kira mentioned, we pioneered the technique where we can target FNDs, those are nanodiamonds, in an intact, cells which are intact in the tissue form, and we can target them. And especially we are having a case where we are able to explain the mechanism of some of these weight loss drugs that's being consumed in the market. The whole idea is that they want to bypass the beta cells in some way, because beta cells are already quite stressed, and they don't want to overstress them. This is where it gave birth to GLPs, glucagon-like peptide. So how do they work, and what is the role of alpha cells sitting in the tissues? That's the question. There's a reason for everything in a tissue, and when you dissociate them and we study individually, largely because of the limitations in the technology, we're losing much of science around nature. So I think there's a lot of work that quantum sensing can do there, and we are proving that. There's also organoids, being built. This is another area. One should know that in FDA.
Sorry,
Yeah.
Deepak.
Sorry, Deepak, you went off to mute.
Oh, yeah, so I was talking about organoids, and then to large degree, FDA has prohibited using animal models in the clinical translation. So organoids are becoming quite evident as an interface for clinical translation over there. And this connects me to 2 points about what is less obvious. One of the work we are doing with organoids is that to demonstrate aging, especially at low gravity aging. So there are a lot of defense reasons for why we are doing it, on this. But the low gravity aging is quite important learning, and quantum sensing basically kickstart that discovery of aging and low gravity. We are working on it, and there's beautiful data set on that topic, and the second thing is, I don't know if you have followed the news a couple of months ago, that in the war in Iran, and the F-36, the pilot of F-36, they were able to point him, somehow. They were able to measure the heartbeat of the person at a long distance somehow. And at least what we think is that there's a lot of knowledge of quantum sensing there, in order to map that out. It's quite hard to get those signatures, at such long distance if it is not quantum sensing. So to just give you an idea of what is less obvious, I mean, quantum sensing has opened up something that a lot of things had hit ceiling, and we are just seeing the beginning, a tip of an iceberg on the topic. Yeah.
That's definitely exciting. I think some of the participants will also have questions regarding the Iran use case, probably. But diving into some of the participants here might not be from a quantum background itself. So, in your research pipeline, where does the quantum part kick in, and how is someone has to be quantum aware or being deep into quantum to join this field? Uh, how is it working right now? Are you focusing on the quantum experts or focusing more on the biosciences expert?
From my side, I'm not a quantum guy, so my background is in mechanical. I did biomedical sciences, but I'm more excited about precision measurement devices, how this, and especially within that domain, sensing is something of topic of interest for me. I worked a lot with capacitive sensors, induction sensors, classical strain gauge sensors. I've touched on different sensing, and I know the limitations of those sensors. And then quantum sensing came in, and then I tried to get excited just because the possibilities it opens up. So, in my opinion, it's not necessary that you need to be a quantum physicist, to do the job, to be in the ecosystem of quantum sensing. Of course, you should know a lot about quantum physics if you are working on certain things like single photon detections. How do you want to do those timing? There are ways on it, and maybe they could play an important role, that knowledge of quantum physics in optical design. And more than anything, in terms of excitation and sensor design itself, like as I mentioned, you use the laser, you can use microwave, and what type of quantum sensors you're going to design. So I mean, NV is only one small part of the big world of quantum sensing. It's ultra low magnetic noise, ultra low gravitational force, ultra low ultra, not ultra low, I say, like identifying temperatures in most critical difficult paths, the pH detection. So there is so many physical quantities that could be detected and monitored over there. So quantum physics will play a role in design of the sensors. But once it is done, somebody has to consume it, you have to productize it, have to build more applications. So in our company, we are around 20 employees at the moment, and we have two quantum physicists actually. Most of them are from the domain of who have PhD, and then let's say in spatial biology, and then who are in the area of mechanical engineering, controls engineering, software. So that's to just give an idea about how we see quantum over there. Yep.
Yeah, Ken, do you want to take this question and also identify some of the gaps you see within the industry and academic space within India?
Yeah, regarding the first point, I mean, very much same thought as what Deepak had. So I am a chemist by training, and I'm an organic chemist. I'm definitely super far off from any quantum phenomenon, right? Um, and I don't think one needs to necessarily be a quantum physicist to work in this space. Yeah, so if you're building applications, if that's the interest that you're in, I don't think you need to be a quantum physicist, but definitely having a collaborator or having a consultant who gives you that input is definitely great. But I don't think you necessarily need to be to develop applications around it. It always goes back to anything that you want to do around science, right? So the science is at the very core of it, and it does not mean that core science is required for every other activity within the organization. So apart from that, there are hundreds of other things that need to work out. So definitely, yeah, you can as long as you have an interest, as long as you have the ability to have a consultant, have a collaborator who can explain those dynamics to you, explain those features to you, you should be good, right? Coming to your second part about gaps, I think, frankly, 2 years ago, if you had asked me, is this a domain that I would be working in, I probably would have nothing that I would tell you because I would not know anything about this domain, right? But in 2 years, there has been a lot around quantum sensing, right? People's knowledge on this area. If you just look at the number of YouTube videos that are out there on quantum sensing, the number of articles, newspaper articles, journal articles around quantum sensing, you will see this massive explosion in the past 2 to 2.5 years. So I think that's an inevitable way that where the gap is kind of closing of what was originally required for the domain and what is available now. And I think in the core sense of the physics of how these sensors work, I think much of the science is pretty well understood. A lot of the theories are very firmly grounded at this point, and I think now is the time for all the application scientists to come in and start building all these layers of applications around this core technique. So in the sense, the gap would essentially be, can you overcome the barrier that this has the name quantum sensing, right? As long as it doesn't spook you out, I think the gap is just not there. But for a lot of people, when I tell my friends, okay, we build quantum sensors and we're looking at applications of quantum sensor, they'll be like, oh, what is this quantum sensor? This must be something really crazy, right? So a lot of this gap uh, is also there because of the term quantum sensing. It does definitely sound very sophisticated and scary, but I think the general market has enough expertise to kind of patch that gap. Yeah, that's I'll wrap that up there.
Yeah. Uh, Another question I had was regarding the policy and governance space. So, uh, as you had mentioned, the technology is moving very rapidly. Are there, is the policy also catching up to it? Be it regarding handling of data, switching from classical to quantum, regarding how the diagnostics would be and how uh, accurate and acute the information would be. Is the policy and regulatory space catching up, or do you see there are some gaps, or do you even see like there's some over-regulation happening?
I I mean I can talk about it from a diagnostics point of view. Uh, See, ultimately it's an ex vivo technique that we are using. We're not putting any quantum sensors within the body and asking a question. So in that sense, the regulations is not a problem for this sort of application where you're taking blood sample out and you're working with the blood sample. Of course, questions will be asked in terms of, are your data consistent? Is your data fidelity okay? Do you see any drift compared to classical data sources? Those sort of questions will always be there, but that's the case for any sort of diagnostic tool. And we are also looking at spaces which have generally not been touched with classical diagnostics. So there's no diagnostic uh, tool to measure stress, right? So the question there will be like, what is the regulatory space to find a new technique for getting a new type of data? And I don't think the regulation will be around the fact that it's going to be quantum sensing, right? So it's independent of the way you measure, but rather what you are measuring, that will be under regulatory purview, right? Because primarily because it's ex vivo. But I don't think from the instrumentation perspective also, I don't think there is any change in regulations. I mean, Deepak can talk more about it, but I don't think there is currently anything that is open for regulation, like something that we see as potentially a government swoops in and says, "Oh, you can't do this anymore." I don't think there is anything like that at this point.
Deepak, you mentioned the case regarding Iran, like how there can be some privacy and anonymity concerns. Are those something that policy folks should be aware about, or is it something that is present in the scientific community, but it's being hyped up by policy?
Yeah, no, I think the definition of policy, we sit in Netherlands, and then there are many European policies that I could just elaborate. So in different areas, So if I see a new technique like quantum sensing, which is at the fringe, not at the center, would the government put a policy in place to enable those ideas to become a conceivable entity in the market, right? So if that's the question, I think that's about the risk appetite of each country on taking a new technology to the market. I think in Netherlands, they have done a wonderful job. I think they started to invest in quantum 15 years ago, you know. And I see that in India, they started to do it 3-4 years ago. The National Quantum Mission. I don't know exactly what it is. So it's a good move, right? So the policies who wants to take that risk and advance that. And there's also policies where, okay, you create this knowledge about the quantum computing or quantum sensing, quantum communications. Those skills are very important for the country as well as for the society, per se. And then the question is that how do you advance their growth? Right. So there are policies in Europe which basically helps you to get launching customers. Like I mentioned about drug discovery, right? I want to go Okay, science is good. The quantum sensing is leaving physics labs, and now I want to go have the first-hand experience of testing it in the market, right? And on the other hand, the market is very they see that it's quite high risk. Why would I take this risk? Things are going normal, and I see only a small changes that I could get by this technology. Their perceptions. There's the government policy has enabled us here in Netherlands to test these cases in the market and demonstrate in the market that is not just a small difference from quantum sensing. It's a magnitude out of difference that you can use to gain value in the company. So there they are. And when it comes to safety and security, I think life sciences is a little bit limited, but there is one case that we know is that quantum sensing can help in some of those bioweapons. So there is a way that the quantum sensing can add value within that area. There the governments could be aware of this and see how quantum sensing, but it's not as big of a security threat or policies like around quantum computing, yet for quantum sensing per se. But in terms of privacy on quantum sensing, it's we don't have much of information. I think it will all go to wearables at a point. Our belief is that as a complex technology like this, in 5 to 10 years down the line, would be packaged into wearables, very, very fast than I could imagine. One of them who's making significant progress on this area is Bosch in Munich. These guys have built the quantum, bulk diamonds that they use it for magnetic cardiography. I think if I MCG instead of ECG. So they're already making, but the question is largely about where data sets, who manages data. But that's classical problem in any measurement technologies about where the data sets and stuff. But I think there is no clear case yet, apart from the Iran that we mentioned, that one has to put more security or safety policies around quantum sensing in life sciences.
At this point, I would like to open the floor to participant questions. I think there have been a few already in the chat box. So one of them had mentioned was, are there any other sensors other than the NV centers, like cold atoms, that are being currently used for quantum sensing and life sciences?
At least from my side, I haven't seen cold atoms used in life sciences because several reasons. I don't know. One, how the physiology of the cells. Whenever you do quantum sensing in life sciences, cells, tissues, or whatever, you should not interfere with the native physiology of the cells. So my knowledge is quite limited when it comes to cold atoms and cells and tissues. So per physics, what cold atoms means by itself, it influences the native physiology of the cells. So I think they're limited. There'll be a lot of limitations even if you want to do. If you want to build an atomic clock, cold atoms do play an important role, I guess. Yeah.
And how is the intersection of AI and quantum computing with this quantum and life sciences helping in characterization or the data processing? Has it increased exponentially as we have seen the increase in AI capability over the past few years, or the techniques used are relatively straightforward and similar to like classical techniques?
I can shed a little bit. So what we are doing with regard to AI is, I told you earlier that we are using quantum sensing as a technique to generate some fundamental data that just never existed all these years, right? So now you have a new data stream that you have, and the question that we are asking is, can I deconvolve all the data in this data stream to give me some sort of a fine-grained image of what's happening around the cell or around the sensor in this case. So let's say there are multiple types of free radicals that are being produced. Can I have curves that are mapped out for each of this type? Then I have a gross cell data. Can I then extract and say, okay, this contribution is from this type of free radical, this contribution is from this, and that would obviously require a lot of computation, a lot of ML and AI data that AI computations that need to be done. That's one place that we are using AI in conjunction with what we are doing with quantum sensing. But ultimately, it boils down to the fact that with quantum sensing, you have a brand new stream of data, and that data is enormously complex. It's got a lot of fine things in it, and we need to tease that data out, and that's what we're using AI for. So that's one place that I'm seeing it. Deepak, anything else that you guys are using it for?
Not we don't do much with AI, but as Kiran mentioned, for us it's we know that the AI companies in drug discovery are interested in functional data sets that we have. There's a new baggage. I mean, not even new weights that they could allocate to dynamic data sets in a cell. But I think there will be areas that will emerge, as I mentioned. In omics, there is already AI, playing a big role. You need to have a big data set, to handle to be able to utilize AI. And I think with quantum sensing, with what we are doing, as the data sets grow, there will be a clear more case for AI to jump in. What we are doing is not AI at the moment, but we want to structure the database as thorough as possible, so that if somebody with AI like Kiran and his team, they would love to deploy some of their models in the future. If they develop, then for us the database structuring for them would be very important. So we provide the data layer, but the AI can come on top of it to create insights of need. Yeah.
Nicholas asked a question regarding the characterization and configurability of the diamond and how the sensor is designed in itself. Do you guys want to take that? How the sensitivity of the hardware itself dictates the application, or the application itself dictate the hardware capability that you get?
Yes.
It's
So
online process.
one of the things, I mean, if you, I think I can break at this point. I can break this down into like, okay, you could have a single NV center in a diamond. Well, it's beautiful. That's more often you will see single NV center diamonds used in metrology. So it's wonderful. One of the examples is the spin-off from Dominic Butke labs from in Munich called Quantum Diamonds. I think also Kasturi, from IIT Mumbai, she works extensively, I guess, on these topics, in metrology, right? So that's one area where more or less the sensors are standardized. The single NV is good. I wouldn't say it's easy relatively to handle. Then you have a little bit more complexity if you make it multiple NV centers in a diamond. Then the orientation of these NV centers will influence your relaxation curve, and stabilizing them gets more complicated. Then I'm going to make it even more complicated now that I'm going to have multiple NV centers in 70 nanometers. So it's not anymore bulk diamond, it's nano diamond, right? So you go down in scale, it makes it more complicated, controlling them and then extracting the data of need over here. So our architecture, the way we have designed it, can handle these, let's say noise layers that comes across here. And then I see the question is about buying them and using them. I think there are many of them in the market who, at the moment, if anybody is interested, I can share something if they shoot an email to me about a different type of sensors they want to use or something. I can guide them to the right places where they could go and experiment on this topic with that. Yeah.
While we wait for more questions, there's a question by Abra. I think we have already touched on it, but what are the form factors and commercialization challenges, especially in the intersection of biotech and quantum sensing? I think we covered quite a bit
Yeah.
on that, but if there's anything that you guys want to add, please feel free.
Nothing from my side on the topic.
Yeah. I think we definitely spoke in length about making it high throughput is something it's a challenge at this point.
Another question I had while we wait for the audience question is, what is the one development in the next 5 years that could fundamentally change your assessment of the field?
I think my opinion is that this is what we also aim for, is that you know, to do multiplexing. You know, we are only looking at free radicals in a cell, but there are many other species of interest that we should be able to measure temperature, for example, pH. You know, for example, there, and at the same time make it faster. You know, so whoever can tap into this at that speed, then they win the hardware level, and whoever can translate this data into insights, you know, instantaneously, they win the data and the AI layer. They win that space. So, and whoever can combine this, they're going to lead quantum sensing, in my opinion. You know, so that's where I would leave it off with that. Yeah.
And also, is there a possibility of using the same sensor and same kind of just one shot sensing for multiple free radicals, or right now is just focused on marking and collecting data on a particular free radical one at a time?
I think from our side, we basically have an NV centers that could pick up any magnetic noise coming around it, you know, and we can't differentiate between hydroxyls, superoxides, peroxynitriles, or, you know, we cannot differentiate them. But what we can do is we can go to the root cause of some of these, for example, mitochondrial RD superoxides, you know. So based on the compartments within the cell, we can target them very specifically and get some readouts on that. But there is a collaboration at the moment. I don't know if it's in the public medium, where you can functionalize the FNDs to read specific free radicals, you know. But there, your controls become very important in order to be very accurate in terms of you're only getting superoxides out of it, or, you know. So the controls and the type of you know, cells or protocols in that way is very important, all in all, for it. But we can't at the moment.
Yeah.
Any other audience questions? We can wait a couple of minutes.
They can also shoot an email, Shantanu,
Yeah.
after, if they need for you. You know, that's also something that that's fine. You know, I can reply to it if later on, if it emerges. Yeah.
Yeah, if there are no further questions, I'd like to give back the time to the speakers and the participants. And thank you so much, Deepak and Kiran, for joining in and for this insightful session.
Yeah. Thanks a lot, Shantanu, and also thanks, Kiran, for being here. Lovely. I learned a lot, and thanks for giving this platform to see, you know, how we can motivate quantum sensing and promote quantum sensing and motivate people to work in that domain. Yeah. Thanks.
Yeah.
Thank you,
so much.
Shantanu. Yeah, thanks, Deepak.
I think one of the participants have asked for the email ID, if possible. If you'd like to share, otherwise we can ask them to that to us. Yeah. Okay.
I just shot my email so they can send it. Yep. Thanks a lot.
Yeah. Thank you, everyone. Thank you for joining in.
Bye bye.